Alright, let's talk about the drug approval process in the US. This will help you understand my part 1 video where I talk about certain GLPs in phase 2 or 3 trials or some that are in regulatory review. Alright, so a drug is developed. What is a drug? As defined by the FDA, a drug is any product that is intended for the use in the diagnosis, cure, mitigation, treatment, or prevention of a disease, and is intended to affect the structure of any function of the body. Step 1 is preclinical, which is animal testing. This is how we test for toxicity. Step 2, a drug manufacturer files an IND application or an investigational new drug application. This is submitted to the FDA, and this is before we start testing on humans. Once that's approved, we start clinical trials. We have phase 1, 2, and 3. Phase 1 trials are with a small group of people to screen for safety. These are with healthy volunteers. This is when we determine safe dosage ranges and then identify side effects. With phase 2 trials, we look at efficacy. Does the drug work for its intended treatment population, or in those that have a specific disease or condition, at the prescribed dose or doses? This is a slightly larger sample. This is with people with a specific disease state we're trying to treat. So we're establishing a therapeutic dose range and then continuing to monitor for side effects. Once all is good, boom, we go to phase 3 trials. This is a pretty large sample size. This is the final confirmation of safety and efficacy. Phase 3 trials still look at efficacy and safety, and then also study different populations and different doses, and then also different drug combinations. A lot of patients are on a lot of medications. I always think it's important with clinical trial data to see exclusion criteria, like who is left out of this trial. It's usually those with uncontrolled disease states like hypertension or diabetes, maybe pregnant people or those with cancer. The FDA then meets with the drug sponsor, and then the drug sponsor files an NDA or a new drug application. So basically a drug manufacturer asks the FDA to market a drug. The FDA has a couple months to review the data and make a decision. I don't think anything happens if they don't approve or deny it within that 60-day period I'll mention. There are also some labeling laws that a drug manufacturer has to abide by. Not some, there's actually a lot of them. The facilities where the drug is manufactured is also inspected, and if all is good, the FDA approves a drug. I will mention a lot of drugs die in clinical development and don't make it to market. It takes about a decade and even longer to get drugs to market usually. There are a couple ways to get it approved faster, like the Accelerated Approval Program or the Fast Track Program. The FDA actually granted Eli Lilly a Fast Track designation for tirzepatide in 2022. It became Zepbound. After a drug is approved for the marketing, we hit something called post-marketing surveillance, or basically phase 4 trials. This is to continually monitor for safety issues after a drug hits the market. The manufacturer is required to submit periodic safety updates to the FDA. Providers and patients can report adverse events to the FDA's MedWatch system. When risks are found, it's added to the drug labeling. One example this year is pulmonary aspiration in patients with GLPs during surgical procedures, requiring general anesthesia or deep sedation. This is despite recommended fasting or liquid diets 24 hours before surgery. GLPs cause delayed gastric emptying. This is kind of how they work. Despite fasting 24 hours before their procedures, some patients might have residual gastric contents. The general recommendation I've seen is not injecting your weekly medication at least a week before your procedure. Or compounded semaglutide from a 503A licensed compounding pharmacy. These pharmacies require patient-specific prescriptions from licensed practitioners. 503A compounding pharmacies have licensed compounding pharmacists and work with FDA-approved suppliers. They must follow USP 797 guidelines for sterile compounding. USP 797 describes the minimum standards that must be met when preparing compounded sterile products. USP 797 is basically what every hospital adheres to when compounding medications in patients. USP 797 addresses compounding definitions, categories, personal training and evaluation, beyond use dates, immediate use compounding, cleaning and disinfecting, facilities and engineering controls, labeling, documentation, quality assurance and quality control, environmental monitoring, including microbial air and surface monitoring, storage, handling, packaging, shipping, and transport of products. Talks about PEC, SEC, ISO Class 5, 7, and 8 environments or clean rooms, and a bunch of other stuff. Basically, compounded semaglutide isn't just made in some guy's basement. But anyway, how long are drugs like Ozempic going to be expensive? When is it going to go generic? Well, it was FDA approved in 2017. How long do drug patents last? Drug patents are usually filed during preclinical trials, and they typically last 20 years. Patents protect research and development investments by blocking a competitor such as a generic manufacturer or another innovator from entering the market with the same or a similar drug product. There are exceptions for compounded products, like I mentioned in my other video, for drug shortages or clinically relevant alterations to drug products. The FDA's main function is to protect consumers from unsafe, ineffective, and fraudulently marketed products, but also to administer regulatory exclusivities to drug companies. So remember, it takes about a decade to get a drug to market, so that patent is usually about halfway up by the time a drug hits the market. With these patents, drug companies can enjoy something called market exclusivity, which prevents any other companies companies from marketing the same drug product. There are ways manufacturers can extend a patent, like with a PTA or a patent term adjustment, or a PTE, a patent term extension. This is a way to compensate for administrative delays with the USPTO or regulatory delays with the FDA approval, respectively. I'll also mention there's something called regulatory exclusivities. Regulatory exclusivities provide parallel market protection for an approved drug, which can extend exclusivity beyond the patent term, or an absence of patent coverage. One example is with pediatric exclusivity. We saw this with Vyvanse. It came off patent technically in February of 2023, but we had market exclusivity of the drug until August of 2023 due to that pediatric indication. 6 months or half a year. Another example is orphan drug exclusivity. An orphan drug is a medication for a rare disease that requires incentives to encourage its development. That's because it's not super profitable. There aren't a lot of people with that specific disease state who are going to buy the medication. An example of an orphan drug is Trikafta. It's for cystic fibrosis. Something that's interesting is a supplemental NDA or an SNDA. A drug company can add an indication to a drug that's currently on the market by submitting a supplemental new drug application to the FDA. They can also submit an SNDA to change an existing drug or label. They can change an existing formulation or even add a new dose or strength. This can add up to 3 years of market exclusivity to a drug. Just to be complete, an ANDA is called an abbreviated new drug application. This is submitted to the FDA for a generic drug approval. These don't require clinical data but must prove bioequivalence. Side tangent, but biologics or biopharmaceuticals are complex pharmaceutical drug products manufactured in, extracted from, or semi-synthesized from biological sources. They have a different regulatory approval pathway because of how complex they are. What's interesting is some GLP-1RAs like Trulicity are technically biologics, but some like semaglutide and tirzepatide are technically not. This is because they don't meet the definition. They're not complex enough or big enough. They're small peptides. This is important because you can't compound biologics under 503A or 503B of the Food, Drug, and Cosmetic Act. I'll also add that insulin was reclassified as a biologic in 2020. Some other examples are monoclonal antibodies like Humira, Orencia, Stelara, recombinant proteins like Epogen and filgrastim, enzymes like tissue plasminogen activator, blood factors, interferons, and many vaccines. The term for generic biologics are biosimilars. An example is Semglee, the long-acting insulin. It's biosimilar to Lantus. It was the first approved biosimilar insulin product. Insulin is a hormone. Another example of another biologic hormone is glucagon. Manufacturers submit something called a BLA or biologics license application to sell a biologic drug on the market. When submitting for approval of biosimilars, remember the generic biologics, they require clinical data. This is a chart from my advanced dosage forms course if you want a difference between like a small molecule versus a biologic product. This is another good snippet from my notes. A random thing on my mind that I'll mention with biologics is something called a biobetter. Biobetters are in the same drug class of an existing approved biologic, but they're not identical to the original and are engineered with some improvements. So a modified version that might have better efficacy, selectivity, or stability compared to the original drug.